Hello friends, welcome to Pivot Breakdowns. In this video, I'll be discussing the structure of proteins. Firstly, let me give you a real-life summary of what protein structure is all about. Now, when we were born, our parents always wanted us to go to school to become medical doctors, engineers, lawyers, and so many other professions. To become any of these professions, the first step was to enroll us for primary school education, also called elementary education. After completing primary school, we obtained secondary school education, after which we got admitted into a university to study the course of our choice. This is called undergraduate education. After completing our undergraduate studies in the university, some of us even went as far as enrolling for postgraduate education, which includes master's program, PhD program, and so on. Now, this is the hierarchy of education, and as we go from level to level, learning becomes more and more complex. So, just like education, the structure of proteins follow a hierarchy or is arranged in levels. The four different levels of protein structure are primary, secondary, tertiary, and quaternary structure. And the complexity of protein structure increases from the primary to the quaternary level. So the primary level of protein structure is the least complex, that is the simplest. And as we move further from level to level, the protein structure becomes more and more complex. Now you can notice the similarity between education and structure of proteins. Primary school education is just like the primary structure of proteins. Secondary school education is just like the secondary structure of proteins. Undergraduate education is similar to the tertiary structure of proteins. And postgraduate education is just like the quaternary structure of proteins. It is the most complex and advanced level. Now, in this video, I will only discuss in details the primary structure of proteins. The other structural levels are discussed in separate videos. So, what is the primary structure of proteins? The primary structure of a protein simply refers to the sequence of its amino acids. Something like this. As you can see, this is the sequence of amino acids in this protein, and so it is its primary structure. Now we know already that amino acids are linked to each other through a type of covalent bond called peptide bond. So when two amino acid molecules combine, a dipeptide is formed. When three amino acids combine, a tripeptide is formed. When four combine, a tetrapeptide is formed. When five combine, a pentapeptide is formed, and so on. However, note that tripeptides, tetrapeptides, pentapeptides, and all short chain peptides, let's say 3 to 35 amino acids long, are generally classified as oligopeptides, since the term oligo means few or small number. And when several amino acids, let's say above 35 to thousands, combine, the product formed is called a polypeptide, since the term poly means many. Also note that the terms protein and polypeptide are sometimes used interchangeably, that is synonymously, since proteins consist of many amino acid residues. So in general, peptides can be divided into two classes, namely oligopeptides and polypeptides. Now, in a peptide, there are two ends, as you can see here. The amino acid residue at the end with a free alpha amino group is called the amino terminal residue, or in short, N-terminal residue, while the amino acid residue at the other end with a free alpha carboxyl group is called the carboxyl terminal or C-terminal residue. So this is the amino terminal end and this the carboxyl terminal end of this tripeptide named alanyl serine threonine, a combination of alanine, serine, and threonine. And every peptide, both oligopeptides and polypeptides, have an amino terminal end and a carboxyl terminal end. Typically, the amino terminal end is the start of a peptide and continues until the carboxyl terminal end. Hence, we usually write the amino acid sequence of a peptide with the amino terminal end placed on the left and the carboxyl terminal end placed on the right. Now, proteins are made up of one or more polypeptide chains, and the sequence of amino acids in the polypeptide chain of a protein is unique to that protein. That is, each type of protein has its unique or particular amino acid sequence. For example, this is the sequence of amino acids in this protein. You can see the amino terminal end and the carboxyl terminal end of this protein. So this sequence is read from left to right, starting with glycine at the N-terminal end. And this sequence or order of amino acids is the primary structure of this protein, and it is unique to this protein. Finally, the uniqueness of the amino acid sequence of a protein is very important as it determines the overall shape and function of the protein. Let me give you an example of how important the uniqueness of the amino acid sequence of a protein is. Now, we all know about sickle cell anemia, a genetic disease of the blood in which the red blood cells have an abnormal crescent or sickle shape instead of a disc shape, something like this. You can see the disc shape of a healthy red blood cell and the abnormal crescent shape of a sickle red blood cell. So what causes sickle cell anemia? Sickle cell anemia is caused by a faulty or defective hemoglobin. Hemoglobin is the oxygen-carrying protein in red blood cells, and it consists of 4 polypeptide chains, that is 2 alpha chains and 2 beta chains, something like this. And each of them has a tightly associated heme group. It is this heme group that binds oxygen. So in sickle cell anemia, the 2 beta chains in hemoglobin have a single amino acid substitution. This is what I mean. Now, these are the first 8 amino acids in the beta chain of normal hemoglobin. This dotted line is just to tell you that the chain continues. As a matter of fact, each beta chain in the hemoglobin molecule contains 146 amino acid residues. I just showed the first 8 here. So in sickle cell anemia, the glutamic acid in the 6th position of the beta chain is replaced with valine. So just this single amino acid substitution changes the overall structure of the hemoglobin molecule and results in the health complications and crisis that comes with sickle cell anemia. Now you've seen how important it is for the amino acid sequence of a protein to remain unchanged. So a change in the amino acid sequence of the polypeptide chain of a protein can alter the overall structure and function of that protein. If you enjoy this lesson, Kindly click the like button and subscribe to my YouTube channel for more videos.